In the last video, we reviewed DNA replication. In this video, we will review the two other key genetic processes, transcription and translation. The nucleic acid RNA plays a much greater role in these two processes than in replication. We will first review the different types of RNA. Each has a unique function and is synthesized by a different type of RNA polymerase. Messenger RNA, or mRNA, is synthesized by RNA polymerase II. It is a copy of the information found in DNA that is sent out of the nucleus to the protein-making machinery in the cytosol. Specifically, it spells out the amino acid sequence of proteins. Transfer RNA, or tRNA, is synthesized by RNA polymerase III. It interprets the message from mRNA and brings the correct amino acid to the protein-making machinery. Ribosomal RNA, or rRNA, is synthesized by RNA polymerase I. It is a key part of ribosomes, the protein-making machinery of the cell. Transcription can be divided broadly into three stages: initiation, elongation, and termination. We will compare these stages in bacteria and eukaryotes. Initiation deals with where transcription begins on a chromosome. In both bacteria and eukaryotes, transcription begins at a specialized DNA region called the promoter. In bacteria, RNA polymerase can bind to DNA directly, but in eukaryotes, other helper proteins called transcription factors must bind before RNA polymerase can. Elongation is the process of growing the RNA strand. In both bacteria and eukaryotes, the enzyme RNA polymerase synthesizes RNA in the 5' to 3' direction. During termination, RNA polymerase and the newly synthesized RNA dissociate from the DNA. In bacteria, termination occurs in a special DNA region called the terminator region. Termination in eukaryotes is still not completely understood, but different types of RNA polymerases use different strategies to end transcription. In eukaryotes, mRNA must undergo processing before it can be exported from the nucleus. Bacteria are simpler organisms and do not need to do this processing. One kind of processing is alternative splicing. mRNA contains exons which code for amino acids, and introns which typically do not code for amino acids. A goal for splicing in general is to remove non-coding introns. The goal of alternative splicing is to create different versions of a protein by splicing out different combinations of introns and exons. Two other forms of processing are the addition of a 5' cap and a poly-A tail. The 5' cap is a modified guanine added to the 5' end of mRNA. It gives the cell a way to control which mRNAs are exported from the nucleus, and it helps position the ribosome at the 5' end of mRNA. The poly-A tail is the addition of several A nucleotides to the 3' end of the mRNA. It helps protect the mRNA from degradation when it is in the cytosol. We will now talk about the genetic code. What is it about mRNA that makes it the recipe for a protein? An important term here is codon. A codon is a set of 3 nucleotides that codes for an amino acid. The sequence of nucleotides in mRNA forms a sequence of codons that codes for amino acids. The table on the right shows the 64 possible codons and the amino acids that each codes for. The correspondence between codon and amino acids is the genetic code. The genetic code is degenerate, or redundant, because multiple codons can code for a single amino acid. However, it is not ambiguous because a single codon does not code for multiple amino acids. We will transition now from talking about transcription to discussing translation, which is the synthesis of protein. First, though, we will take a closer look at tRNA and the ribosome. tRNA is folded into a clover shape and at one end contains an anticodon that base pairs with the codon of mRNA. However, there is flexibility in the base pairing of the third codon position. This phenomenon is known as wobble and explains the redundancy of the genetic code. Multiple codons can pair with a single anticodon, So, multiple codons can code for a single amino acid. Enzymes called aminoacyl tRNA synthetases use ATP to attach amino acids to the tRNA with the correct anticodon. Ribosomes are the protein-making machinery of the cell. There are 3 sites in the ribosome that are involved in protein synthesis. The A site is the site for an incoming tRNA with an amino acid. The P site is for the tRNA holding the growing polypeptide chain. The E site is the exit site, where used tRNAs can exit the ribosome. The webpage here contains a thorough animation of translation, which you can view at your own pace. From the animation, you should come away with a fundamental understanding of the steps involved in translation. We will review these steps briefly here. Translation starts with initiation, in which the ribosome scans for the start codon AUG, which codes for the amino acid methionine. The elongation phase is a cycle. First, new amino acids attached to tRNA enter the ribosome at the A site. Then, a peptide bond forms between the polypeptide chain attached to the tRNA in the P site and the newly arrived amino acid in the A site. Then, the entire ribosome assembly shifts, causing the old tRNA to exit and the new tRNA with the polypeptide chain to now be in the P site. The last stage of translation is termination, which occurs when a stop codon is reached. When this happens, a release factor binds to the mRNA and causes the dissociation of the polypeptide. Translation ends when the ribosomal subunits dissociate from the mRNA.